The overall goal of this novel workflow is to determine the occupancy or stoichiometry of protein post-translational modifications at specific acylation sites. This novel mass spectrometric workflow provides a higher level of precision of quantification.
This method can help answer key questions about reversible protein regulation by post-translational modification, such as which acetylation or succinylation sites are most highly modified and may alter protein function. The main advantage of this technique is that site-level stoichiometry of lysine acetylation and succinylation are accurately determined, and the method can determine stoichiometry from peptides with multiple lysine residues.
This technique is highly relevant because it reveals acetylation and succinylation stoichiometry, or proportion modified versus unmodified, instead of measuring a dimensionless relative abundance change.
I first had the idea for this method when I realized that using data-independent acquisitions and MS2-based quantification for stoichiometry analysis will greatly improve measurement accuracy.
Prepare 3 replicates of 100 micrograms of bovine serum albumin, or BSA protein sample, in parallel at a concentration of 1 microgram per microliter using urea and TEAB solution. Reduce 100 microliters of BSA sample with 8 microliters of 250 millimolar DTT to reach a final concentration of 20 millimolar DTT. Vortex each sample briefly, and incubate the sample at 37 degrees Celsius for 30 minutes with agitation at 40.